Great, good afternoon everybody. Thank you to Nicholas and to Tor for the invitation. This was originally billed as a talk by Klaus Enno Stefan. He unfortunately couldn't make it today, so I am the substitute teacher, but also developer of DCM. So hopefully what I'm going to do today is bring you from A to Z through the vanilla DCM, which is deterministic and bilinear dynamic causal model for task-based fMRI. And so to outline the roadmap, what I'd like to introduce is all the different pieces that sandwich together to produce this methodology for effective connectivity analysis, and start with some basics, and we've seen in Eugene's talk an overview of the different pros and cons of the different methods of connectivity analysis. So I won't dwell on it too much, just set the scene to put DCM in the context of these different other methods that you can use. And then I'm going to just go through and painstaking detail, maybe, the neural level model, just so everyone's on the same page as to what the actual causal inference is about. Again, that came up in Martin's lecture. And then go on to the hemodynamics that are modeled as well in DCM. And then finally put the piece together, which brings from a generative model, it brings us to an analysis framework using Bayesian, variational Bayesian techniques, so we can invert our models given real data. And then I'm going to show you two applications of DCM to fMRI, so just so that you can think about maybe your own data and what you want to model. And I'll use for this the classic Christian Buchel data set, which we move around SPM for different kinds of road tests, which is the attention to motion in the visual system. And then I'll bring you something a bit more contemporary from a group at the Donders Institute who were looking at synesthesia and how DCM could unwrap some of the hypotheses proposed for that interesting phenomenon. Okay, so as we know, and this is why we're all at OHBM, so there's a large and strong history of functional specialization literature in functional neuroimaging, and also arising from centuries-old lesion studies. But a complementary approach to looking at the functional specialization, and they really do go together, is to study functional integration in the brain. So over different timescales, but also, as you'll see in DCM, we can understand different contextual manipulations of networks. So for example, if you're trying to perform some working memory task during the day, where you have lots of nice neuromodulators from the subcortical system sending and enhancing synaptic efficacy in the cortex, the connectivity might change slightly as we're about to fall asleep and have perhaps less excitatory neuromodulation from brainstem regions to cortical networks. And so it's this kind of context dependency that is well captured by the model, particularly the simple model that I'm going to show you today. And it really isn't complicated. And just to put it in context, these kind of overview articles are kind of nice. Kerry Smith wrote one in Nature in 2012, just celebrating fMRI and its 20-year history, and noted in the paper that in the rise of fMRI, that connectivity is now perhaps outnumbering the number of studies published in comparison to the older brain activation studies. And you've already seen two of these images before, I think today, or at least reference to them. And these were the kind of some of the classic connectivity analysis methods and papers. So Bharat Biswal's resting motor cortical networks, which look a lot like, so they're on the left in B, or on the right in B, they look a lot like the task-based functional correlations that you see in motor networks in the image in A. And then other ways to look at connectivity, again, as Eugene summarized, was to perform things like independent component analysis and look at different resting state networks like default mode and task positive and task negative analysis, again, using things like ICA or functional correlations. And this, I think, is just a nice illustrative dichotomy and an outline of nomenclature by Olaf Sporn's Scholarpedia entry. And here you can see outlined on the macaque brain, and Nicholas mentioned it in the introduction, three different types of connectivity. When we talk about brain connectivity, we may be referring to structural connectivity, so the presence of axonal connections, which we can measure with invasive tractography studies or diffusion tensor studies. Functional connectivity is, and this has been again outlined in the previous talk by Eugene, really the assessment of statistical dependencies between time series. and it's mechanistic free, so you don't have to really pin a mechanistic model underneath your analysis, though it doesn't often give you directionality. So you may use, for example, a simple correlation in a seed-based region approach to look at the different connections in, for example, hierarchical visual cortex, is shown here in the middle. What DCM belongs to is the class of effective connectivity models, and it comes from Arston, the idea or the concept of using the minimal circuit model to describe the propagation of activity through circuits. And so, by virtue of the fact that you have a model, you can, or you have to propose directionality in a dynamic sense or in a causal, a control theory causal sense, where we're examining the directed influences between neurons, or mainly neuronal populations in the case of dynamic causal modeling. And as I said, there's an explicit parameterization that you put into these models, which are all assumptions, so you have to try and make them as viable and biologically plausible as you can. And again, it's something to consider when you're looking at different ways of analyzing your data. And so these effective connectivity methods include things like structural equation modeling, dynamic causal modeling, and certain types of Granger causal analysis. So work by Anil Seth's group in Brighton have shown the autoregressive modeling techniques and how Granger can look like DCM without parameterizing in certain directions. And so essentially we break functional and effective connectivity into mechanistic free versus a mechanistic underpinning to the inference you're about to make on your data. Just to mention, and maybe useful for some of the developers in the group, DCM is modular, so we have and it's applied in different imaging modalities. And so generally it's a framework, and if you're developing code, there might be some pieces that you can borrow or plug in to your own particular model for a certain system. And so just very generally, at a big overview, what we model in DCM is essentially a multi-input, multi-output, or MIMO state space model, where at the level of the brain, we have neural state equations describing the propagation through real neuronal populations. And of course, we can't measure this, so this is what we call the hidden layer. But what we can measure is its influence on measurable signal, like levels of deoxyhemoglobin and the resultant bold response. And so in fMRI, we have a relatively simple neuronal model with only one, or at the moment a maximum of 2 states describing the activity in a given neuronal population. Whereas for some of you who do multimodal imaging, EEG and MEG models can get a bit more detailed as a result of their higher temporal resolution. Okay, but today we're talking about DCM for fMRI, and so luckily this morning has lined this up beautifully. So we've seen the physiology, and Rasmus has brought us through some of the intricacies, particularly in the hemodynamic response, that are interesting from the point of view of DCM in terms of second delays in different HRFs in different regions of the brain. But just to summarize our typical paradigm, but also the whole generative pipeline that DCM attempts to model, and again, Eugene mentioned that they use DCM to do bold generative signal data for their analysis of other connectivity measures earlier. So the idea is that we have some stimulus, which leads to changes in neuronal activity and changes in neurovascular coupling, which produces this hemodynamic response, which we can detect in our MRI scanner. And so for DCM, and this is in its original deterministic context where we tell it the inputs that we're giving it, we might have some timed perturbations. So for example, you might be asking your participants to encode scenes for a memory test, for example. This is of Hawaii, Paradise here, on the left. And this will, these particular timed perturbations will be recorded by us, the experimenter, because we know then what's leading to certain unobservable interactions in neural dynamics. And so DCM takes the input and models a set of predictive outputs based on how you connect different regions of the brain. And then, as I mentioned, we use a forward model, which is a static mapping from the neural perturbations to the observed hemodynamic response. Okay, so at the neural level, as I said, it's a multi-input, multi-output, so standard engineering-based dynamic model. And it's really not at all complicated in terms of the assumptions that we might make. If we're thinking about DCM for fMRI for the moment as a psychologist or experimental neuroscientist using imaging, we might motivate the DCM as what I'm going to just tell you. And in a couple of slides, I'll show you how you could motivate it as an engineer who knows nothing about the brain, for example. But for the moment, let's presume that we know that we have 3 regions active in under given task manipulation. We'll call then activity in these different regions X1, X2, and X3. And what we're measuring in DCM is the evolution of the activity, so the temporal derivative or the state changes. And we assume that the changes in the model in deterministic DCM are determined by the current state of the neural activity in that region, but also external inputs like those pictures that we're giving, which can go into multiple nodes, and again, this can be tested, as I'll show you later on. And we presume that the changes in activity is dependent on the connectivity theta. So we have connectivity controlling the activity. We also have the activity controlling the evolution of the activity, as well as external inputs. And they don't necessarily have to be picture onsets and offsets or auditory onsets or offsets. They could be more subtle contextual changes like risky or not so risky decisions, for example. And so at the bottom of our DCM, so going all the way up, we're going to start with a simple linear differential equation, dx dt, which is some function of the states x, also of the external inputs, and the connectivity theta, which controls the strength between the different regions and also within the region. And just to give you a more fleshed out example, so if we wanted to say present things to view in the left visual field and the right visual field, and we get entries into the right and left lingual gyrus, and that we see in our activation that we also have some fusiform gyrus activity, then we could model, for example, the right lingual gyrus x2 dot as having interactions from region x1, which is on the left, and the strength of that interaction is going to be controlled by this connectivity parameter A21. We also think there might be some top-down activity, which we'll call A24, which is the activity coming in from the right fusiform gyrus. We also, as I mentioned, have in DCM control of dynamics within a region. So this is by virtue of the fact that in DCM for fMRI, we're looking at neural activity within an area. And so as you saw today in some of the talks, you could be looking at a group of voxels and typically you extract some summary of that for DCM. And so often what you see is a dampening of a particular region by its own activity. And so that we're going to encode by the parameter A22. And then finally, this particular region gets input from the left visual field called U1. And that strength, the strength of that input will be controlled by one other parameter, C21. And so we can go on and enumerate that same dynamic description for each of the 4 regions. So for example, the right visual field could be getting a different input, U2. And again, for X3, or the rate of change in the left fusiform gyrus and the right fusiform gyrus. So this is just a quite generic way of describing how activity might change in 4 brain regions dependent on inputs that you're sending into visual cortex. And so just this is to get you into the nomenclature of DCM, and again, this is useful, so all the notation that I use today is actually what's encoded in the little MATLAB files that you'll be optimizing and using for model comparison and parameter reporting. And so what we call all the states X, and then we can break this equation into matrix form where we have state changes and then a large A matrix, which we call the effective connectivity. And also, so these are obviously produced by changes in the system states X, or they control changes in the system state X. And also we can include input parameters C, which control the strength of the external input U1 and U2. So you should be able to see how that little dynamic in that 4-region model is represented here by, and again, we saw these connectivity matrices in Eugene's talk, where we have the diagonal describing self-connectivity and the off-diagonal components describing connectivity from, say, X3 to the impact on the rate of change of X1. So altogether, we'll have this simple linear ordinary differential equation where x dot is equal to ax plus cu. Now, what we often do is add one more level of nonlinearity, so it's technically a bilinear addition, where we could presume that there's some change in connectivity that's directly under control of an input. And so this, for example, is given here, and we'll follow this up later on, by the modulation of certain connections by a third input, which we call attention. So we're presuming that the subject is in the scanner, and at some points they're attending to some aspect of the design or your stimuli, and at some other points they're not attending. And so what we're going to do is use another matrix, what we call the B or the bilinear term, to describe the influence of the connections on each other when this input is on or live. So if you look now at the expanded equation, if we, for example, look at the equation for x1, we can see that the rate of change of x1 is dependent, again, like before, on its own connection, A12, but also we can see that there's this new B12, which is the attentional input offered by U3, turning on more connections from U2. So this connection here is going to be modulated under attention with an additive part, B. Okay, so that's the kind of vanilla DCM. It's x dot is equal to A plus the sum over inputs of B times x plus CU. Now, if you were to not think about a subject in a scanner and what areas might be connected or illuminated under certain task conditions, we could motivate the bilinear state equation for DCM by just being an agnostic engineer and using a Taylor series expansion where we're looking at the first couple of terms to describe the activity. Obviously, this is an infinite series, so if we used all terms, we'd have a perfect reconstruction of our signal. But we could assume that we're just going to use the Jacobian A, which is basically the flow or the deterministic flow of the states at a particular resting state, which we use as 0. So this is why it's deterministic. If there's no input into the system, we assume that all the states are at 0 and nothing changes. So we have to give it some driving input to get the dynamics kicked off, if you like. We also use the C parameter in this expansion and the bilinear expansion term, which is the B matrix, which is the partial derivatives over states and also inputs. So it's essentially the effect of certain regions on other regions and on itself under the context of some controlled experimental perturbation. Okay, sorry, wrong way. So what do these parameters mean? Well, if we look at a very simple DCM, so just a one region DCM which we'll call activity here x1, and it has a self-connection A11, then we can write out the full DCM equations at the neural level as dxdt equal to A11x1. And we can solve it analytically because it's fairly simple. And this is your high school differential equation solvers. So what we're getting is an exponential function where A11 is acting as a rate constant. So just to draw that out, what we typically have in DCM is a prior that these self-connections are negative. So this will prevent any runaway dynamics or epileptic dynamics, if you like, in our network. And we see that A11 is the decay function that determines the half-life. So by the time x1 has dropped to half of its initial value, x1 at 0, it's in a time that's proportional to A11. So it's inversely proportional with this constant here, which is the natural log of 2. So the consequence of that is that the parameters are measured in hertz, so they're a measure of, they're a rate measurement, and also to interpret not self-connections, but connections between regions, we can say that if A to B is 0.1 hertz, this means that the rate of increase in the activity of B corresponds to 10% of the current activity in A. And so we don't do an analytic solution for every DCM. What we use is approximate methods like many of you would use when doing dynamic models. So we use a kind of variant on Runge-Kutta. But essentially the parameters of connectivity are rate constants that control the rate of activity in one region relative to the region that is connecting to it. And just to give you an example of a time series, we might have a stimulus occurring going into this region, and you can see nicely like a little delta function that occurs. This is not a very good randomized design, but just to illustrate what the activity looks like in x1 under a particular self-connection A11. So we have this self-connection A11, and you can see that means that the activity dies down rather than exploding every time you have a new input. And then we might make our network a little bit more complicated and add a second region and look at the dynamics in the two-region network. And here what I'm describing is essentially the attention modulation that I showed you before. And so we might have activity in X2 that's under the influence of region X1. And so you can see it's not getting direct inputs like X1 is. And so its activity is a little smaller. But it does respond with some magnitude because of this connection A21. And then we add on a context dependency, which shows us that the neural response here, we're not at bold yet, so the neural response gets a little larger when the manipulation of U2 is high. And so that's just a feeling for how the temporal dynamics at the neuronal level operate. But we know that for DCM, we include, very importantly, a hemodynamic model, which I'm going to show you next. So what I've described thus far is the direct inputs that we get from our GLM, or experimental design, the modulation of connectivity under the influence of some input, which we'll call matrix B, and also the endogenous connectivity, so the stuff that responds or that is present in the network and sends signals in response to input C. Okay, so the hemodynamic level, we know that the cognitive system is modeled at the neuronal level, as you've just seen, but it's in essence unobservable. And so the observable that we do get is the neuronal dynamics transformed into area-specific bold responses. And so the reason that we include this hemodynamic model in DCM for fMRI is to account for the variability that Rasmus alluded to, and also that Eugene mentioned about the idea of temporal precedence may not always be entirely valid in an fMRI setting where the vasculature may be causing consistent delays. And again, there's ways around this with clever experimental design. But this was an interesting paper, if any of you are interested, by Olivier David, who showed in rodent electrophysiology in both fMRI and with spiking measurements that he looked at the validation of DCM for fMRI to look at signal propagation, which was where we had a ground truth of electrophysiology, and showed that using temporal precedence measurements, just in this particular case, the two regions he was interested in, amygdala and thalamus, had slightly different AHRFs, and so making inference just on the bold response itself was giving you false results. So the Buxton balloon model is what's used to convert neuronal activity here at the bottom up into the observed bold response in DCM. And I won't go in detail through the changes in vasodilation, inducing changes of blood flow and changes in deoxyhemoglobin, but we have parameters that are in the model where we have fairly good priors on what they are, things like Grubb's exponent and hemodynamic transit times. And so we put these parameters into the forward model from each of the regions. So the neural activity doesn't affect, from other regions, doesn't affect this output, bold output, but it's just the neuronal activity from the given region transformed through these region-specific balloon models that give us the measured bold output. So finally, what we're getting is a Y, which represents the simulated observation of the bold response plus some noise. So what we're going to see is, again, our stick functions or delta functions showing us when particular events happened. And in blue, you can see the very small neuronal dynamics that we were modeling earlier. And then in red, you can see the simulated fMRI data based on typical hemodynamic response functions, which again, as I said, are optimized for each region individually for DCM. So the first thing that people often think when they see that slide is, okay, there's a lot of parameters in DCM, so how are you able to fit all of those parameters without having parameter interdependency? And this is an image from class, and he showed the posterior correlation matrix. So this is just the normalized posterior covariance of the different parameters. And essentially, the important thing here to note is that the hemodynamic parameters are listed here from 15 to 45. Whereas the really key neuronal parameters, which we're often interested in reporting and testing, are shown up here in the top left-hand corner. And you can see that the co-explained covariance amongst the hemodynamic and the neuronal parameters is quite low. So there is quite a bit of correlation amongst the different parameters in the hemodynamic model, but between the hemodynamic and neuronal model in this particular data set, there's a nice separation of experimental or data variance so that we can make good assumptions on the neuronal level without contaminating it, if you like, by correlations from the hemodynamic level. And this is an empirically testable thing in each of your data sets. Okay. And so the last piece, so we have our neural model and then our hemodynamic model, so we're able to generate bold data. But what if we have bold data and we want to go back and look at the parameters? And that's where we come to the Bayesian piece. So this is Bayes' rule, which simply says that the probability of something theta given some data y is proportional to the likelihood of that data under the particular parameter theta, and also multiplied by the prior probability p theta. So what it allows you to do is incorporate prior knowledge, things like those hemodynamic parameters, but also in DCM it just allows you to do a good model comparison. And so this is just Bayes' rule in pictorial form. And it's important to note that most of the priors in DCM for fMRI are uninformed when we're talking about the connectivity parameters. So we typically put them at 0. So they would have a prior mean at 0, and the data would have to drag it either positively or negatively away from 0 in order to make an inference that a certain parameter has a connectivity from region A to region B. Overall, what we're using is variational Bayes, as I mentioned. And so what you have to do before you get to your Bayesian inversion piece is just specify what kind of noise you think is in the data. And what we use in DCM is the noise assumption is Gaussian, but we have temporal autocorrelations along a particular time series. So this is typically true for fMRI bold responses where you don't get absolutely independent samples at every TR. So we include some temporal autocorrelations and then assume a Gaussian noise response so that we can do our probabilistic inference or inversion. I can maybe take anyone interested in this through this offline because I'm running out of time, but the VB approach essentially is, rather than sampling, what you do is presume a partition of your parameter space and update each one in a kind of expectation maximization setting. And what we're using as the gradient, or the cost function rather, is a term called the free energy, which incorporates data likelihood and also complexity. And the partition, so the Bayesian piece where we look at two different parameter settings, is the noise versus everything in the neural and hemodynamic response. So when you look at, if you dig into the code in DCM, you'll see that in a function called SPM NLSI, so nonlinear system identification Gauss-Newton, you'll see a gradient descent scheme that optimizes the sufficient statistics of the large multivariate Gaussian parameter space, which incorporates all those connectivity and hemodynamic parameters, and then you separate out the parameters of the observation noise. And we can assume they're relatively independent. We assume observation noise is really a property of the scanner and the data acquired, whereas the other parameters should be independent of how the signal is being observed. Okay, and so just to go through that in a nutshell, we have regional responses, and we're going to specify some generative model with different connectivities between regions. Then we do a variational expectation maximization algorithm, which is an iterative procedure which computes the model response under the current set of parameters. It compares the model response with data, and it tries to improve the parameters, if possible, using a gradient descent on free energy. And from that estimation, what we're able to obtain is two important things: both the posterior distribution of parameters, but also the model evidence, which is another common way to compare models without actually reporting parameters. We're just going to look at the evidence of a particular model. Just to show you briefly how you do this reporting, and there are a number of different ways you can do it. If you just did a DCM on a single subject, you can just look at the probability distributions when you've seen the data, so the a posteriori estimates, and you can ask, for example, if their probability mass is much greater than 1 and report those numbers. You may also want, in a group setting, to do a fixed effects Bayesian analysis, and I'll bring you to the key equation here. It's just a simple weighting of each of your participants' posterior mean with their posterior precision or inverse variance. So therefore, if you have 20 subjects and you wanted to report just one parameter averaged over those subjects, what you could do is take a Bayesian approach and look at the goodness of the estimability reported via the precision of the posterior estimate, and then report just one number for your group. Or we can take a classical kind of second level approach and take every mean estimate from the posterior, take your particular parameters of interest and do things like a t-test. So is one connection bigger than 0 across my group of subjects? Or are certain parameters different in my group? Or indeed, we can do things like ANOVAs and test for patient effects if you like as well. An alternative way to report your DCMs and to really do model hypothesis testing is to look at model comparison. And so this is just an illustration of what a good model is. It's something that accurately captures the data without fitting noise, essentially. And so I have 2 minutes, so I'm going to go super quickly. So the punchline is that we can use the cost function and its final output to do this comparison of accuracy versus complexity. Okay, and so there are some numbers in the literature, so you can report now Bayes factors, which are comparable in these numbers for weak, positive, and strong, giving you log odds ratios of, say, 20 when we're in this range. So we have something in Bayes factors that gives us a kind of feeling for the number like you would a 0.05 p-value in classical statistics. And so it might be useful in your papers just to reference the Cass and Raftery paper from 1995, which gives, for people who aren't familiar with Bayes factors, I mean reviewers. Okay, and I won't go through this, but we can look at this again if anyone's interested offline in the particular measure of complexity. It's suffice to say it's different to the ACAC information criteria or the Bayesian information criteria. It's a little more involved, accounting for parameter covariations. Okay, I might get you through one DCM for fMRI. Let me skip this one for the moment, because if any of you have been to the SBM courses, you've seen this analyzed to death. And so instead, I'll just bring you to an interesting study on synesthesia. So this was done in, as I said, at the Donders Institute, and I just picked it as an interesting example of model hypothesis. So the team were looking at synesthetes. Specifically, they had grapheme color synesthesia, where people saw particular graphemes, like the letter C-O-L-O-R, in different colors. And so they got an impressive number of participants for this study, and they asked whether changes in effective connectivity can explain synesthesia activity in V4, because a lot of these synesthetes had activations in the color region of the brain when they saw non-colored letters. And so there was two hypotheses that were interesting to the team, whether there was direct cross-activation in ventro-occipital cortex. And so that was essentially a model where they had the word form area here, or the letter shape area, sending connections directly to V4 and then into parietal, superior parietal lobule. Or they had a disinhibition feedback hypothesis, where rather the letter shape area sent a forward connection to the superior parietal lobule, and that in turn sent a backward connection, and that's why you were seeing activation in V4. So they were their two interesting hypotheses on synesthesia. And initially there was no winning model, so they tested the two models and couldn't find a winner. But they went back to their subjects and did a difference between people who projected their synesthesia or associated their synesthesia. And projectors were people who reported directly seeing the color in a letter in the visual field, whereas associators saw it in their mind's eye. So they didn't directly see the letter O as yellow, but they just thought yellow. And then when the team went back and reanalyzed their DCMs, they found that the model separated according to the type of synesthete that they were testing. And so what they found was that the projectors had greater evidence for a cross-activation model where LSA directly fed signals into V4, whereas the people who associated had the activity due to these grapheme experiences. It seemed to be a superior parietal lobule effect onto V4. And interestingly, they were also able to correlate the strength of model comparison with the particular sensory experience of their participants, where people who were more associators or projectors had greater model evidence than those who were somewhere in between. Okay, so just to wrap up, so DCM isn't one specific model now at this stage. There's lots and lots of developments, post-hoc model optimization. You can now do actually stochastic resting state studies in DCM, which we'll save for another day. But it's useful to report the DCM version that you're using in your papers. And also, in some way, it's really not a lot different from a GLM, except you're looking at the dynamic activations between regions. And so your studies are designed basically exactly the same way as you would to do a good GLM analysis. And as I said, there's lots of new developments, not a lot of time to get through them all today, but I'll just thank the organisers again, and Klaas and Tanika, who was involved in that synesthesia study, and Mohammed Seeger and all the Phil Methods group. Thank you very much.